Thank you for joining today's Coffee Break webinar. Today's topic is the derivation and characterization of peripheral neurons. Human pluripotent stem cells provide a powerful platform to model diseases of sensory neurons in the peripheral nervous system and study their underlying mechanisms. Unfortunately, protocols to differentiate sensory neurons from human pluripotent stem cells can be lengthy, technically difficult, and may not yield large quantities of the desired cell types. In this webinar, Dr. Nadja Zeltner and Dr. Kenyi Saito-Diaz of the University of Georgia describe a versatile protocol to differentiate sensory neurons from human pluripotent stem cells. Dr. Zeltner completed her PhD at the Axion School of Medicine at Mount Sinai in New York, working on the use of adeno-associated virus for gene therapy. After working in the Studer Lab at Memorial Sloan Kettering Cancer Center, Dr. Zeltner joined UGA as an assistant professor in early 2018. Dr. Saito-Diaz completed his PhD at Vanderbilt University, where he studied the molecular mechanism of the Wnt signaling pathway. In 2018, he joined the Zeltner Lab at UGA as a postdoctoral research associate. Together, they will describe how differentiated neurons go through the appropriate developmental stages, taking advantage of these stages to enrich for specific sensory neuronal subtypes. Welcome, Dr. Zeltner and Dr. Saito-Diaz.
We are interested in studying the peripheral nervous system, which comprises all the nerves outside the brain and the spinal cord. They transmit stimuli from the environment to the central nervous system. The peripheral nervous system is divided into sensory and autonomic nervous system. There are three main subtypes of neurons within the sensory nervous system: the nociceptors, which detect pain, the mechanoreceptors, which detect touch, and the proprioceptors, which detect the body's position. Diseases of the peripheral nervous system can be hereditary, such as familial dysautonomia or familial diverticulitis, and others. They can also be acquired through diabetic neuropathies or neuropathy induced by anti-cancer drugs. The induced pluripotent stem cell technology is a powerful tool to study the peripheral nervous system. In this system, we can isolate somatic cells from patients and reprogram them into iPS cells. We can then differentiate these iPS cells into a pool of sensory neurons that comprises all three subtypes, or we can isolate each subtype. For example, we can isolate nociceptors to study pain or hereditary diseases such as FD. Then, using this platform, we can identify potential drugs which could be used as a treatment for patients. One example of a hereditary disease affecting the PNS is familial dysautonomia. FD is a genetic disease caused by a mutation in the gene ELP1, which causes its downregulation. FD affects development and survival of all the PNS, including autonomic and sensory neurons. Patients show a wide range of symptoms. For example, a symptom caused by dysfunction of autonomic and sensory neurons is the dysautonomic crisis, which are vomiting episodes that occur when the patient gets excited or stressed. On the other hand, due to loss of sensory neurons, patients have decreased pain and temperature sensation. Currently, there is no cure for FD. There are only palliative treatments, and patients rarely live past 40 years old. Thus, there is a clear need to study and understand FD in neurons of the peripheral nervous system. As I mentioned before, the iPS cell technology is an ideal tool to study peripheral neurons. However, a critical step to characterize iPS cell-derived peripheral neurons is to assess their functionality. Multielectrode arrays are an excellent platform for these types of studies. In our studies, we used the Axion Maestro Pro because it's easy to use and allows us to measure the activity of a network of neurons as well as to manipulate the environment, for example, using small molecules, and assess their effects in the firing of action potentials without killing them.
To model diseases of the peripheral nervous system, we adopted a protocol to differentiate pluripotent stem cells into sensory neurons using chemically defined conditions. As you can see, the cells pass through the developmental stage of neurocrest prior to differentiating into sensory neurons. Here you can see that neural crest cells are positive for the transcription marker SOX10, whereas sensory neurons are positive for the pan-neuronal marker QJ1 and the sensory neuron-specific marker BRN3A. Furthermore, this protocol results in an increased differentiation of nociceptors marked by the surface marker of track A, compared to mechanoreceptors and proprioceptors marked by track B and track C respectively. This composition is similar to what is observed in the human dorsal root ganglia. To test whether the sensory neurons are electrically active, we turn to the MEA Axion Maestro Pro. During differentiation, we replated the neural crest cells into MEA plates, continued the differentiation, and monitored their electrical activity over time. Here you can see a representative image of the plating of our sensory neurons in a well of the MEA plate. The black lanes indicate electrodes. In this heat map, you can see the differences of firing rate between day 20 and day 50 of differentiation. Furthermore, you can see that the sensory neurons start firing on day 25 and continue to fire up to day 80. Additionally, the number of action potential bursts peaks at day 65 as well as the firing frequency. Finally, we use 3 different nociceptor agonists, capsaicin, Win5512, and pregnenolone, which all increase the firing rate of our sensory neurons. Finally, we developed 2 methods to select for specific neuronal subtypes. First, we use immunopanning to isolate individual subtypes using antibodies against the surface markers track A, track B, and track C, which are expressed in nociceptors, mechanoreceptors, and proprioceptors respectively. Second, we changed the date when we replaced the neural crest cells to push the differentiation towards mechanoreceptors. We tested the functionality using the Axion Maestro Pro. We plated the mechanoreceptors in the MEA plates and measured their firing rate under normal conditions. We then incubated the sensory neurons in hyposmotic media, which would induce pressure to the sensory neurons, mimicking touch, and measure the firing rate. As you can see, hyposmotic media significantly increased the firing rate, suggesting that the mechanoreceptors are functionally active. With this study, we were able to efficiently generate sensory neurons from pluripotent stem cells, including induced pluripotent stem cells generated from patients. We were able to obtain a pool of neurons containing nociceptors, mechanoreceptors, and proprioceptors, which can be used to study development and different aspects of diseases of the peripheral nervous system, including axon regeneration. Furthermore, we were able to isolate specific subtypes, which can be used to study their biology and potentially identify new small molecules that could be used as potential treatments. Here, we presented a protocol to generate sensory neurons using chemically defined conditions and are electrically functional. We also demonstrate 2 distinct methods to enrich for a specific sensory neuron subtype, which are also active. We hope that this protocol will be a platform to study development and diseases of the peripheral nervous system, specifically of sensory neurons, and hopefully this knowledge will help us develop new treatments.
And that is the conclusion for today's Coffee Break webinar. If you have any questions you would like to ask regarding the research presented here, or if you are interested in presenting your own research with bioelectronic assays, please forward them to coffeebreak@axionbio.com. Thank you for joining in on today's Coffee Break webinar, and we look forward to seeing you again.